looked at the, oh, never mind. I'll catch you guys later.
Are you sure? All right.
Hello.
He's on.
Having a
I'm gonna hold you to
conversation
that.
before I got here, huh?
Yeah.
Oh yeah, we were talking. Yeah, about
Okay.
questions.
All right.
Okay.
Before we begin, are there any questions?
Well, I guess we were just kind of talking about that whole second grading of the labs. I was just looking at lab 00 and lab 1, and the end date is July 22nd. So do we have until July 22nd to get our corrections back to you?
For lab 00, you do have until July 22nd to get your corrections back to me. For lab 01, I've only got about half of the students graded, so I'm going to extend the due date, and it'll probably be from a week from this Saturday. Okay? And it'll be a little confusing because they're put in the same folder, but anyways, that's what I was planning to do.
Awesome. Thank you so much for clarifying that.
Okay. Some people I've gotten lab 01 graded, but some people I have not. I mean, I think I have all of you graded partway, but I don't have it graded all the way. But I might be mistaken, and that is I only have half of you graded. Anyways, I will extend that because not everyone is graded. Let me share my screen here. Come on. There we go. Did any of you notice I started a discussion group? Let's see if I can find that. I think it's in the bottom of the general course information. Student discussion. So if you ever want to discuss things with other students, you want to interact with each other, you can ask questions, you can answer questions, you can form a study group in this. This is just a place for you guys to comment, ask questions to each other. I am not going to generally look at this, and I will not answer questions. I may, if I notice that somebody answered a question and their answer is wrong, I will then comment. But this is a place that I'm getting for you guys so that you guys can interact with each other. And I know some people are trying to form a study group. This would be a good way to contact people. The other good way is you need to email everybody in the class, and you can do that just by going to the people link and then get their email address. I don't know if you can email everybody.
Yeah, our girl Cassie, she emailed everybody. She came
Yeah,
to
but
collect.
you can do that. I don't know, like I said, I don't know how you can do that, but you can do that. You may have to do it one after the other. If you find my email done to everybody in the class, you could get it that way, except I think I've got it blind, in which case I don't know if you can email everyone. You can open that email and then see if you can email everyone. So that might be another way you could do it. But like I said, this would be a good way to get in contact with people. And it's just designed for you guys to interact with each other. Okay. Any question about any of that? So this is your space. You guys do with it as you like. I will not generally answer questions here. Unless, like I said, I won't answer the question, but I'll make a correction on somebody else's answer. If you ask me a question, you should email me. Don't put it in the student discussion area. Okay. Any questions about any of that? I have the first couple of textbooks in the textbook. First chapter is working. If you want the later summaries for the textbooks, please let me know and I'll try and get them working. I don't know why, but when I rolled this over, all of the links went to something like spring 2022. And I have access to that class, so I can get it, but you guys don't have access to those classes. And I don't know why this didn't roll over in the normal way, but it didn't. And so these links aren't working. But these links up here are working. And that's for chapter 1, chapter 10, chapter 2, and chapter 3. These are complete pages to all of these chapters. All of these others are just the chapter outline and then the clinical case study. Any questions about any of that? And thank you for the student who pointed that out to me. I was not aware that when I rolled this over that these links weren't working. Anything else I need to talk about? Like I said, I'm a little behind on the grading. Let me stop my share and then I can tell you what I graded and what I haven't got graded. I really worked hard on getting the, I guess I'm not sharing anymore. I really worked hard in trying to get the lab 01 graded, but it's just taking me too long. We're a fairly large class, and whenever there's an error, I have to say what's wrong and give a hint to how to grade it. So like I said, it's taken me much longer than I'd hoped to get lab 01 graded. But lab 00 is graded, and you must get the corrections for the second grading in by this Saturday for lab 00. I'm trying to see the worksheet here. Now you're not seeing this, right? Can somebody confirm you're not seeing?
Correct. I can't see your screen.
Right.
Yeah.
I'm bringing up the grade book to tell you what I've got graded and why isn't that displaying? I believe I've got the first lecture assignment graded as well. I have not graded the first quiz, first practice quiz. I have to manually go in there and grade it because it's supposed to be worth 0 points. So every question is worth 0 points. And then any points you get will be extra credit points. And so I'll give you something like 0.1 or 0.2 credits for each correct answer. Oh, I've got a lot of pictures. That's good. I have, oh, look at that. I've got worksheet lecture 1 graded, but I haven't put in the grades yet, so it's not fully done. And worksheet lecture 2 is not due until this Saturday, so I don't have that one graded yet. Worksheet lecture 1 is graded, and if you don't have a grade in your score, it'll be full credit. Okay, I mean, I haven't gone back and put in the full credit. That was my mistake. But I'll get that done. All right, that's it for the grading. Any question about any of that? Let me shut down the grade book and now I'll re-show my screen. Come on. All right, you should be seeing my screen now. So what we're doing today is July 20th. We are supposed to be finishing chapter 2 and starting chapter 3. We are behind schedule and I'm not sure we'll get there. Today in the lab, we have lab 03 on microscope basics. Is that right? Yeah, lab 03. Any question about any of that? All right, if not, let's move to chapter 2. Chapter 2 is a chapter on chemistry. And for many years, I asked the primary microbiology instructor to get rid of chapter 2, that it's just covering material that you guys had in your preliminary biology class. However, I now have the opinion that you guys need this review of chapter 2 and that you guys actually struggle with chapter 2. It's not a hard chapter, but students seem to not fully understand chemistry. And so I'm in favor of keeping chapter 2 in this course. All right, we talked about the acid-base balance and how the pH scale works from 0 to 14, and how it's actually the measure of the hydrogen ion concentration in solution. And the pH is exactly equal to the negative log of the hydrogen ion concentration. So let's moving to organic compounds. You're seeing organic compounds here. Let me blow this up so you can see it a little better. They can be somewhat simple. Acetic acid is a simple organic compound. There's actually methane, which is the simplest. These are both simple, so is ethane. And they can be complex molecules too. This is much simpler than a protein. Let me get that out of the way. This is much simpler than a protein, but it's a pretty complicated molecule. When we show chemistry, you can draw out the chemical bonds like this or like that, and that's a stick figure. You can also have the ring figure shown here. Wherever there's a point, that means there's a carbon, and the carbons and generally the molecules are not shown. If ever you draw it out in the ring structure and it's not a carbon, then you have to put in the element there. And let me see if I can blow that up. All of these are carbon, so I'm not sure what point it'll be. I can't seem to blow it up. But you have to put in what the element is if it's not a carbon in the point. In the ring structure, you have double carbon rings shown with a double bond, meaning, come on, mouse. This is one bond right there, and there's one bond there. So these carbons are linked together with 2 carbon bonds, which we don't seem to have one with a carbon, but there's a good one, showing you a double covalent bond between carbon and oxygen. All right, any questions about any of that? Organic compounds contain carbon. They also contain hydrogen. If you look at all of the organic compounds that we showed you here, and then all the others down here and wherever, they always contain carbon and hydrogen. The simplest organic compounds are called hydrocarbons. And that's all they contain is carbon and hydrogen. And that would be something like methane gas or ethane or propane, isobutane, if that's what that is. I can't read it. Okay, simply carbon and hydrogen. These might be carbons and hydrogens. I'd have to blow it up to be certain though. So hydrocarbons are simple organic compounds. There are other organic compounds which are more complex, and they may contain other elements. They oftentimes contain oxygen, although there may not be much oxygen in an organic chemical compound. Let me see if I can blow this up again. Here we go. So in this molecule here, there are actually only a couple of carbons, oxygen right here, right here, right here, right here, right here, and here. So in this entire molecule, there's only 6 atoms of oxygen. But like I said, they can, well, minus the phosphate group, let's put on another hydrocarbon right there, and then we won't have those 4 oxygens there, but so oxygen can be present, it doesn't have to be present. And when oxygen is there, it's usually in much less numbers than the carbon and the hydrogens. You'll note that the organic compounds tend to be held together by links between carbon. Let me blow that up again. Like right here, all the links are held together by carbon. Most of the links here are also held together by carbon, although oxygen is holding the link right there. And there, carbon's holding the molecule together. So carbon is said to be the backbone of an organic molecule. What's holding the molecule together by the covalent chemical bonds. There may be other elements present like nitrogen, sulfur, and phosphorus. All of them are held together generally by the carbon. And nitrogen, sulfur, and phosphorus are usually present at low numbers. Compared to inorganic molecules, organic molecules are more complex, and they're capable of more complicated biological functions. Here we're going to talk about some examples. Methanol is an organic chemical compound, a little more complex than methane. And obviously I've stated that the molecule holding them together, the molecules together are generally the carbon bond. So the carbon bond backbone is called the carbon skeleton or the carbon backbone. There may be functional groups. In fact, in our organic chemical molecule, there almost are always functional groups attached to the molecule. The functional groups are important because they give the molecule different properties than the molecule would have without that functional group. For example, where do I have that shown? Right there. Methane shown here is very similar to methanol shown here. The only difference is the hydrogen is replaced by an OH, but that OH gives this molecule different properties than methane. For example, methane is a lipid-soluble molecule, nonpolar, and it does not like dissolving in polar molecules such as water. So it generally separates from water. Methanol, on the other hand, is polar in this region, non-polar everywhere else, but polar in this region, and it can dissolve in water. So the functional group changes the properties of the molecule. And here we're seeing OH as a functional group on 3 different molecules. Let's talk a little bit more about the functional group. They're responsible for most of the chemical and physical properties of the particular organic chemical compounds or molecules. And the biological molecules have different functions depending on their functional groups. So hydroxyl group, OH, in alcohols is hydrophilic. It attracts water and helps the molecule dissolve in water. Some other functional groups that we're going to talk about are NH2. This is called the amino group, and it's found in proteins and amino acids. Another one we're going to talk about is carboxyl COOH, and that first O has a double covalent bond to it. This is called the carboxyl group. It's found in proteins, amino acids, lipids, and organic acids. And then there's phosphate, P O4, and notice how it has a negative charge to it. It's found in ATP, DNA, and RNA. Why you need to know the groups which are shown in the red boxes is because these are the functional groups that are important in biological molecules. Any question about any of that? Frequently, you can find more than one functional group found in a single molecule, like an amino acid, which is a pretty small molecule, has the amino group on one end and the carboxyl group on the other end. And then down here is just a hydrogen. And then up there, it's the R group, which is the group that determines which amino acid this is. And R just stands for anything, and it can vary depending on which R group we're talking about, or which amino acid we're talking about. Any question about any of that? All right. For the biological molecule, they can be small organic molecules, or they can be large organic molecules. The small organic molecules can combine to make the larger macromolecules. The small molecules are called monomers, and the macromolecules, or the large molecules, are called polymers. A polymer means that it consists of many small repeating molecules called the monomers. For example, starch is a macromolecule and a polymer, and it consists of only glucose molecule, one after the other, linked together. And that's all starch is. Any question about that? And glucose is a monomer. You can join smaller molecules together to make a larger molecule by dehydration synthesis, also called condensation reaction, by pulling off OH from this monomer and H from this monomer, and then directly linking the R's together. So R is linked together, and then the water comes out, and the OH and the H form together to make water. And so water comes off of this reaction. Why it's called dehydration synthesis. You're making something, so that's synthesis, and then water comes out, and hydration means water. Any question about any of that? You can also split a larger molecule like this polymer by adding water, and then the reaction goes this way in hydrolysis reaction, and then the water splits apart, and part of it joins this part, the H joins this molecule, and then the OH of water joins the R on this molecule, and that is called hydrolysis. You should be familiar with hydrolysis if you've ever watched the movie Back to the Future. And I think it's Back to the Future 3, but it could be Back to the Future 2. I don't remember. It's where Marty McFly travels to the future, and he sees himself or something like that, where he's older, and he comes to visit his mother, and his mother sticks a hockey puck into her hydration machines, and then she runs it, and the hockey puck changes into a pizza, and then she serves it to the family. And Marty McFly says, "Ma, you sure know how to hydrate a pizza." So his mother was doing hydrolysis. Any question about that? All right, this is showing you in longer form. If you have a polymer and you want to add a monomer to it, you can undergo dehydration synthesis, and OH comes off the monomer, H comes off the polymer, and then this monomer will bind to that polymer, making a larger polymer, and then making water also. You can also undergo hydrolysis, having water come in, splitting off the monomer. And the water breaks apart with the H forming the smaller polymer and the OH joining the monomer. So hydrolysis and condensation reactions are the reverse of each other. All right, let's talk about the carbohydrates. The first of the four biological molecules. There are four biological molecules, and the four, you can find three of them on any box of cereal or any box of food. One is carbohydrates, two is proteins, and three are lipids, although on the box of cereal it won't go lipid, it'll call it a fat. And then the fourth biological molecule you do not find on a box of cereal or any other food that I'm aware of, and that is nucleic acids. Carbohydrates include sugar, starch, and other complex carbohydrates. And then by sugar, there's many different types of sugars, and we'll talk a little bit about that. Carbohydrates are an important energy source, so we consume carbohydrates mainly to get our energy needs, meaning when we eat food, the first food that'll be digested to make our energy needs, meaning make ATP, will be carbohydrates. And it'll first use glucose, and then when all the glucose is used up, it'll start digesting the complex carbohydrates like starch. It will not use other foods to make the body's energy needs until something like 45 minutes into a marathon runner's run. So the marathon runner has to be running for 45 minutes before the marathon runner will start digesting lipids to make their energy needs. Any question about that? Carbohydrates consist of carbon, hydrogen, and oxygen. And I should mention that all of these biological molecules are an organic chemical compound, but they're also one of the four biological molecules of life. I guess I should mention why we call them the four biological molecules of life, and that is because you only find these molecules in something that is alive or was once alive. Meaning if you go rock digging and you find a fossil and you discover that there's a biological molecule in that fossil, it's because that fossil was once alive. Okay? If on the other hand, you go looking through the soil of the moon, you will not find a biological molecule because there's never been life on the moon, unless you put it in a spacesuit or something like that. Obviously, astronauts have gone to the moon, but there's never been life growing on the moon. Any question about that? All right, carbohydrates consist of carbon, hydrogen, and oxygen, but it's not just any arrangement of carbon, hydrogen, and oxygen. They actually have a formula. All carbohydrates are made up of formula. For one carbon, there are two hydrogens and one oxygen. And then for the carbohydrate, you can just take this formula and then multiply by a number, and then you'll get it. Like, for example, glucose is carbon, hydrogen, and oxygen, and then you multiply by 6. So the glucose is C6H12O6. Any question about any of that? All right, simple sugars or strings of simple sugars are simply linked together to make the larger biological molecule. We can classify the carbohydrates into three major groups based on their size. The smallest are the monosaccharides, and they include the simple sugars. And then they are the disaccharides, and that's simply two simple sugars bound together, like lactose, the sugar found in milk, is a disaccharide. Glucose is a monosaccharide. A polysaccharide like starch is a very large molecule, and it's made up of simply glucose repeating one after the other. No idea how many glucose molecules are in starch, but it's a large number. Okay, many more than 10, probably more than 100, but I don't know. I'd have to look it up. Any question about any of that? All right, let's talk about the monosaccharides, the simple sugars. All of the monosaccharides are made up of 3 to 7 carbon atoms. However, most of the monosaccharides are made up of 5 or 6 carbons. The monosaccharides are sweet molecules, meaning they sweeten our food, and they are water soluble, so they will dissolve in water. The disaccharides are formed by joining 2 monosaccharides in dehydration synthesis, so you can get glucose shown here and fructose shown here, pull out water in dehydration synthesis, and then you get sucrose shown here. If you don't know, sucrose is table sugar. Glucose is blood sugar. Fructose is fruit sugar, if you don't know. That's not terribly important, although you should know sucrose is probably table sugar. And then water comes out. You can then break down the sucrose, which happens when we eat table sugar, and water comes in, and then hydrolysis occurs of the sucrose, and then the sucrose breaks into glucose and fructose. Any question about any of that? All right, the polysaccharides consist of many monosaccharides that can be straight or branch chains. Starch is the simplest of the polysaccharides, and it's mostly a straight chain molecule of nothing but glucose held together, one after the other, and it's many glucose. It's not 6 here, which is shown. It's just many glucose held together. Some examples of polysaccharides include starch, which is found in plants. Many plants store glucose as starch. The exceptions are sugar beets and cane sugar or sugar cane, and it stores the glucose as sucrose or table sugar. And that's why we get sugar from sugar beets and sugar cane. Glycogen is how animals store glucose. We store glycogen down in our muscle cells, and most of it is stored in our liver. Dextran and cellulose are all glucose polymers. And chitin is also, but we'll talk about, we're not going to talk about chitin, so don't know it. And here you can see some starchy foods. Everybody knows bread, cereal, pasta, rice, and potatoes are starchy foods. A lot of people don't realize beans and chestnuts are also starchy foods. Beans are about 1/3 carbohydrate, 1/3 protein, and 1/3 fat. But they are a large and good source of carbohydrates. And chestnuts are also, and I don't know the chemical makeup of chestnuts, so I'm not going to state it. If they had a peanut there, I could state it, but I don't know for chestnuts. This page is showing you some complex polysaccharides. You do not have to memorize this table. I'm just trying to show you some complex carbohydrate. Let's put that there because chitin we're not going to talk about. Starch, mostly a straight chain where glucose is just repeated one after the other. Glycogen is very similar. All it is a chain of glucose one after the other. However, glycogen is a little different in that it has the chain branches. I can't blow that up any. I tried. So the glycogen branches. Cellulose, which is found in grass and other plant matter, all it is glucose held one after the other in a chain. I need to blow this one up because that's not showing well. And this, let's see if I can blow it up here. Sort of. So the blue line of cellulose is where cellulose is held together by a covalent chemical bond. And cellulose is just row after row of sugars held together in a covalent bonds. And then those covalent bonds, or the rows of covalent bonds, have hydrogen bonds between the sugars. And if you notice, there are a lot of hydrogen bonds in this row here and that row there. It looks like every sugar from here to there is hydrogen bonded to the neighboring sugar. And then in this row, it's even more because it's not an external one. It's binding to this row and that row with hydrogen bonds. Any question about any of that? If you recall, we talked about what a hydrogen bond was in our lesson yesterday. It's simply where the hydrogen of one molecule is attracted to, in this case, the oxygen of another molecule. And hydrogen can also hydrogen bond with nitrogen. It doesn't have to be with oxygen, but in this case, it's probably with oxygen because this is cellulose. There isn't nitrogen in cellulose. The link is a little different than the link of starch. Oops, went too far. But you don't need to know that. And then later, we're not going to talk about it now, but later in the term, we'll talk about peptidoglycan, which has, oops, too far. Oh, shoot, moving the wrong one. Which does have carbohydrates linked together one after the other. It also has short peptides in the peptidoglycan, and we'll see that later. There would be a short peptide hooking this chain to that chain. Like I said, don't worry about peptidoglycan. We'll talk about it later. All right, any questions about carbohydrates? Come on. If not, let's talk about the second of the biological molecules, lipids. Lipids consist of carbon, hydrogen, and oxygen. However, most of the lipids consist of carbon and hydrogen. Let me show you that. For example, in this lipid here, it's mostly carbon and hydrogen. All of the molecule here is nothing but carbon and hydrogen. The only oxygens are right here and then right there, what this is going to bind to. And that will, I think, break off, although it might break on to an OH, I don't remember. This is showing you fatty acid. a major component of lipids. The lipids can be a small polymer, which is made up of monomer. And fatty acids would be one of the monomers which makes a lipid. Fatty acids are a major component of lipids. They consist mainly of chains of carbon and hydrogen bound to it. It does have a carboxyl group at one end, and most other locations it's just hydrogen bonded to the carbon. With fatty acids, we have two types. This one here is a saturated fatty acid. That means all of the carbon-carbon bonds have a single link, a single covalent link. There's no double covalent link. There are also unsaturated fatty acids, and to be an unsaturated fatty acid, all you need is one of the carbon links to be a double covalent bond, where there's going to be a double covalent bond, or the unsaturated fatty acid, where that is, will cause a kink in this hydrocarbon chain. And I think that's shown in the next slide here. Yes, right there. So let me blow this up. So right there, you can see the double carbon bond, and all the other carbon bonds here in this molecule, as well as all of them in here and here, are single covalent bonds. And you only need one in the entire molecule to call this lipid a unsaturated fatty acid or unsaturated fat. Okay? If there are no double carbon bonds, then it's a saturated fat. And it's kind of surprising that there's very little difference between this molecule here, which is unsaturated, and then you make that a single carbon bond, and how you do that is you bind two hydrogens there and there. Oops, go back. It's kind of surprising that unsaturated fats are so much better for the heart than a saturated fat. But when a saturated fat is circulating in the blood, it is not a good thing for the heart. And if you have too much of it, it will increase your risk of getting a heart attack. And then an unsaturated fat will reduce your risk of getting a heart attack, which is why people should eat things like olive oil and other forms of oil, because they are unsaturated fats. Any question about any of that? All right, this is a picture showing you a simple lipid. It is made up of fatty acid moiety, shown here, and then a glycerol moiety, shown there. And for most of the fats in our body, we have 3 fatty acids which bind to the glycerol. And when they go through dehydration synthesis to do that, water comes out and OH comes off of the fatty acid portion, and then a hydrogen comes off the glycerol. So that the molecule will then look like this, where this one only has one oxygen, and then this OH group has switched to oxygen, and then that oxygen binds directly to the carbon. The most common fat in your body is a triglyceride, shown here, and a triglyceride is a fat molecule that simply has 3 fatty acid groups attached to the glycerol. If you want to see a triglyceride molecule, just look down at your belly. You've got lots of triglycerides there. Any questions about that? All right, let me state that the body's richest energy source comes from fats. So if you wanted a bunch of energy from a food source, let's say you're stranded in Point Barrow, Alaska in the wintertime, and you have to walk 20 miles into the town because your car is broke down and you want to carry what food you can, but you don't want to carry more than 10 or 20 pounds because it's going to get too heavy and then you get tired and then you won't make it to Point Barrow. So what you should take with you to eat is actually a fat, because you'll get the most energy out of fats than any other food. And we're talking about pound for pound. You'll get more energy out of a pound of fat than you would out of a pound of carbohydrates or a pound of protein or a pound of nucleic acid, which nobody eats that, but I just thought I would mention it. That doesn't mean it's the best thing to eat, and it certainly wouldn't necessarily be healthy for your heart, but it would get you the energy you need to hike 20 miles in the wintertime to Point Barrow, Alaska. Any question about any of that? All right, another lipid we need to talk about are phospholipids, and they do have, let me blow this up. They are a monomer. They do have glycerol shown right here, and they do have two fatty acid chains attached to it. And then they have a third molecule attached, and it's a monomer, and it's the phosphate group containing phosphate, and then an R group, and R can stand for any organic molecular group. Phospholipids have this type of a shape where they have a head, which contains the phosphate group and the glycerol, shown here. And this part of the molecule is polar. Phosphate is polar. A glycerol is slightly polar, although in this configuration, it's not very polar. With the OH on it, it would be polar, but that's not very polar, as it is CO. C is not very polar. And then on the other side of the molecule, it has the fatty acid moieties, and they are nonpolar. That makes this phospholipid a biphasic molecule, where this part of the molecule can dissolve in water, and this part of the molecule can dissolve in lipids or oil. There are not that many biphasic molecules, but you are aware of one of them, and that is soaps and detergents are biphasic. And that's why they clean better than water alone. The soap has part of the molecule which can dissolve in water. It's not a phospholipid, but I'm just using it as my point of discussion here. So the detergent or soap has a part that can dissolve in the water, and then it has a part that can dissolve in oil. The part that dissolves in oil helps us clean our hands and our clothes, because this will pull out the oil from our clothes, and then that will make this be dissolved in the water of the washing machine. So that's why detergent helps clean our clothes. The water can get out a polar molecule from our clothes, but the water will not remove a nonpolar molecule. But the soap or detergent can remove the nonpolar molecules from our clothes. And that's why we use detergent or soap to clean our hands or to clean our laundry. When we get a bunch of phospholipids shown here with the head and the tails, and we put them in water, they'll actually start lining up this way, with the heads facing out towards the water here, and the heads facing out towards the water. And then the tails do not want to be around water, and so they will go to the inside. This is showing you the membrane of a animal cell, and phospholipids are the main molecule in a membrane. And animal cells have cholesterol in the membrane to help give the membrane strength, because this membrane without the cholesterol would be pretty weak. But phospholipids are the main molecule in animal cells' membrane. It's just that animal cells have cholesterol also in the membrane. And we do that because we do not have a cell wall. And so our cells need a little bit more strength than the phospholipid membrane can give it. If you don't know, cholesterol is only found in animal tissue. It's never found in plant tissue. And that's because plants do not need to add cholesterol to their membranes. They have a cell wall, and the cell wall will protect the membrane, the cell membrane, much better than cholesterol will by giving the membrane some strength. Any question about any of that? All right, I started to say that when you put fatty acids in water, they'll start lining up like that. They'll actually start lining up like this when you put this phospholipid in water. And that'll be the phosphate head is facing outside, which is going to be the water. And then there can be water on the inside. And we have heads on the inside facing the water inside the membrane or the phospholipid bilayer. And what does this remind you guys of? What does that look like? Come on, somebody's got to have an idea. Well, think about what that looks like. It's kind of important, but I'm not going to say if nobody's going to guess.
Cell membrane.
Yeah, that looks like the membranes of a cell. And this actually is the concept of a cell, where inside the cell, we mainly have water. Sorry, my mouse is dying. Mainly have water, and that's because the cytoplasm is mostly water. And then we have the membrane, which will be a phospholipid double layer like this. And then outside the cell, we have the heads facing outside, because outside the cell is mostly water. Think of a cell in blood. Most of the blood will be water. In reality, wherever the cell is in the tissue, most of outside the cell will be water, because that's what we're mostly made up of. But in the blood, it's clearer. All right, there are some other lipids we need to talk about. There are steroids and sterols. Steroids are 4 interconnected carbon rings characteristic of this group. So 1, 2, 3, 4, and wherever there's a point, there's a carbon. And then it has this attached to it. And then the OH is attached to it for cholesterol. If ever there's an OH group attached to a steroid, we call it a sterol. So cholesterol is a sterol. And as I mentioned, we have cholesterol in our cell membranes. Any question about that? Let me see if there's anything else I wanted to say about lipids. The phospholipid is a very different lipid, because all other lipids do not dissolve in water. The phospholipid is the only lipid, at least in the body, that dissolves in water. And it also dissolves in oil. So phospholipid is a biphasic molecule. There was something else I needed to talk about here. I talked about cholesterol. Steroids and sterols are not monomers and polymers, meaning the steroid is not made up of repeating small units. And so for lipids, many of the molecules are monomers or made up of monomers. And you can call them polymers, but they're really small polymers. Like a phospholipid only has 3 parts attached to the glycerol. So that means there's only 4 monomers in phospholipid. Here, let's go there now. 1 monomer is the phosphate group. Excuse me, there's only 3, no, there's 4 monomers in the phospholipid. 1 being the phosphate group, 3 and 4 being the fatty acid tails of the phospholipid molecule, and then the 4th being glycerol. Those are the monomers of phospholipid. I should mention that this, and then when we talk about this molecule, that this is the head, and then the tail is the fatty acid portion. All right, any question about lipids? If not, let's talk about proteins. Proteins are essential in cell structure and function. There are many different proteins, and they can function differently in cells. Some proteins are structural proteins, and they are found in like hair and collagen, structural protein, bone and muscle. And so these proteins we call structural proteins. And they help the muscle move, or they hold something together, like in bone, they help hold the bone together. There's also enzymes, and enzymes are biological catalysts, speeding up a chemical reaction. There are transport proteins, and these are molecules that move other molecules across the cell membrane, either into or out of the cell. There are regulatory proteins, and they regulate a body function. For example, a regulatory protein in a child would be something like epidermal growth factor that helps the child grow. There are proteins that are responsible for movement. The reason why all of our muscles move is because of a protein that contracts when our muscles move. And it's the contraction of this protein that causes the muscle to move. Proteins are also responsible for some bacterial toxins. We'll talk about toxins later in the term, but something that is a toxin is something that is toxic to us. And some bacteria make a molecule which is toxic to us. And that would be a bacterial toxin. I should mention that not all toxins in bacteria are proteins, but the majority of them are. And then there's finally antibodies, which are protein molecules that the host makes in order to defend itself. So the antibodies can go and bind to antigens that have invaded the host. You can see that proteins are very diverse in their function, and they are the most diverse of all the biological molecules. Why they are so diverse is because most of the other biological molecules are made up of one or a few repeating monomers. I don't know how many simple sugars there are, but there's at most a handful, okay? And that's at most. Most of the time, our bodies only see glucose and fructose and beta galactose. Most of the time, our body does not see other monomers. Any question about any of that? All right. So let's talk about proteins. Proteins are a molecule consisting of a monomer called amino acids. And the reason why proteins are the most diverse of the biological molecules is because they have more monomers than any other biological molecule. For example, humans have 20 different amino acids which get together to form the proteins. For lipids, we only have a few different monomers which can form the lipids. For nucleic acids, this one I know. For DNA, we only have 4 different nucleotides to form the DNA. But for proteins, humans have 20 different amino acids. To get a protein, you simply link many different amino acids together. There are 20 different amino acids. Each of them has a different R group. The simplest is glycine, the amino acid glycine. And for R, shown here, all R is a hydrogen atom. Alanine is fairly simple amino acid too. It has a carboxyl group, CH3. No, a methyl group, sorry, a methyl group attached to it. You don't need to know this table, so do not memorize it. I'm just trying to show you that you vary the R group to get the specific amino acid. And any amino acid that has a CH3 in the R group is the amino acid alanine, at least for humans. Let me go back a minute. These R groups are all hydrophobic, meaning they do not like being around water because they're non-polar molecules in the R group. In the amino group and the carboxyl group, these are polar, but the R group is non-polar. We can have R groups which are polar, like tyrosine has an OH group attached, and that changes this molecule, which we'll show over here, from a non-polar group to a polar group. Histidine is a polar amino acid, and then tryptophan is slightly polar. It does have one polar group right here. Any question about any of that? Amino acids are linked together by a peptide bond. What you do is you just pull off water in dehydration synthesis, and then you directly link this carbon to that nitrogen. That bond is fairly uncommon on the planet Earth and in the universe, where carbon bonds directly to nitrogen. It does happen in proteins, which are peptides, and that's why we call it a peptide bond. Any question about any of that? Oops, didn't mean to move forward. Once you link all the amino acids together, and there's going to be no more amino acid added, once that peptide or polypeptide has added its last amino acid, we no longer call it a polypeptide or a growing protein. We now call it a finished protein. So it's a protein. Before that, it's a polypeptide. Any question about any of that? So strictly speaking, you should never call a protein that's being synthesized. You should never call it a protein because it's not a protein yet. You have to add the last amino acid before you can call it a protein. There are different levels of protein structure. And let's watch a little video on that. Not seeing it. Let me do it directly. Yeah. That's not what I wanted. What do I want? I want that. Well, that was a chore. Looks like we have to watch a commercial first.
You and your family deserve to feel safe online, and McAfee can help protect your...
In this video, we're going to talk about the structure of proteins. And what basically is a protein? A protein is a polymer consistent of many amino acids. So each amino acid represented by the circle is a monomer that forms the protein. Whenever you have many amino acids, it's also called a polypeptide. And the bond that connects each individual amino acid residue is a peptide bond. So let's talk about the individual structure of an amino acid. So an amino acid has a chiral carbon with a hydrogen attached to it, and it has an amine group attached to it as well. And then it has an R group and a carboxyl group. Now I'm going to draw another amino acid right next to it. The left side, where the nitrogen is located, is the N-terminal of the amino acid. The right side, that has the carboxyl group, is the C-terminal of the amino acid. Now, these two amino acids can react with each other in a condensation reaction where we're going to lose water, and we're going to connect the two amino acids into one molecule. So once water is removed, we could form a peptide bond. This is also called a dehydration reaction because you're losing water. And so this is going to be the new product of the reaction. So let's see if I can fit it in here. So this oxygen is gone. So now we have a nitrogen. Let me take out H2O. We have one hydrogen left over. We lost two once we lost water. So this is a dipeptide because we have two amino acids combined. And so this amide bond that we see here, that is the peptide bond. We formed a bond between the carbon and the nitrogen atom. So whenever you combine two amino acids to form a peptide bond, you're forming a covalent bond, which is hard to break. Now let's talk about the different levels of protein structure. There's 4 levels that you need to know: the primary structure, the secondary structure, the tertiary structure, and finally the quaternary structure. Now the primary structure is simply based on the sequence of the amino acids found in the protein. The sequence determines the shape of the protein. If you replace just one amino acid with another, it will completely change the shape of the protein. So the shape and its function is primarily determined by the sequence of amino acids. The secondary structure describes the localized shape of a protein. And there's two of them that you need to be familiar with: the alpha helix and the beta pleated sheet. In the alpha helix, you could see that it's stabilized by hydrogen bonds. The NH group of one amino acid interacts with the carbonyl group of another amino acid. And each turn contains about 3.6 amino acid residues. Now here is a visual illustration of the beta pleated sheet. Just like the alpha helix, it too is stabilized by hydrogen bonds between the carbonyl group of one amino acid and the NH group of another amino acid. Next up, we have the tertiary structure, which represents the three-dimensional complete folding pattern of the protein. And so here's a visual illustration of it. So we could see some areas is just a straight chain. Here we have an alpha helix, and here we have a beta pleated sheet. Now, the tertiary structure is one individual subunit. When you combine multiple subunits, you create a quaternary structure. So hemoglobin is an example of that. It has 4 individual subunits, 2 alpha subunits and 2 beta subunits. So hemoglobin is a protein with a quaternary structure. And so that's basically it for this video. Hopefully it gave you a basic understanding of proteins and the 4 levels of structure that they have. Thanks for watching.
If you live in Washington, you could lower your electric
Okay.
bill to less.
Any questions about that video? So there are 4 levels of protein structure: primary level, the secondary level, the tertiary level, and the quaternary level. The primary structure is just the sequence of amino acids in a polypeptide chain. So one amino acid following another is the primary structure. Once a protein is first made, all it has is the primary structure. However, the nearby amino acids in a protein start to react with each other, and some of them will be attracted to other nearby amino acids, and some of them will be repulsed, and that begins the secondary structure. Now, your textbook, for reasons I'm not sure, and some of the web say that the secondary structure is only occurring by hydrogen bonds. That is not correct. The secondary structure is any localized interaction between neighboring amino acids. It can be a hydrogen bond, like if this red one hydrogen bonds to this blue amino acid, but it can also be an ionic interaction. So if this, I guess that would have to be a hydrogen bond because it's the same. Like this one here, the orange one and the green one, if this is a positive charged amino acid, the green one is a negative charged amino acid, we're going to have an attraction right there between those amino acids. Positive charge will be attracted to a negative charge. And then you can have a repulsion. If this yellow amino acid is positively charged and this yellow one is positively charged, these 2 amino acids will be repulsed from each other. You can also have repulsion between a polar amino acid, like this yellow one, and a non-polar amino acid, like this pink one. So that would be a repulsion right there. That's different than the ionic repulsion. Let's see. Oh, there's one more attraction, and that is van der Waals forces. Non-polar molecules like to be together. And if you put like a drop of oil in a bottle of oil, the oil will converge. And that is from van der Waals forces. where the non-polar molecules want to be together. They are attracted to each other. And there is actually a physical attraction. Like that's why when you put oil in oil, meaning drops of oil in oil, it all stays liquid. It's staying a liquid because those non-polar molecules are all attracted to each other. And that is why it is a liquid. If the molecules were not attracted to each other and were repulsed from each other, then that would not be a liquid container of oil. It would be a gaseous container of oil. I think that's all the attractions. The attractions can be hydrogen bonds. They can be hydrophobic interactions, like a repulsion between a polar molecule and a non-polar molecule. There can be repulsion between two positively charged molecules. There can be attraction, ionic, between a positive charge and a negative charge. There can be attraction between hydrogen bonding. And these are the secondary structure. The secondary structure only happens between nearby amino acids. And it happens between neighboring or nearby amino acids. It does not happen from an amino acid at the start of the molecule and an amino acid at the end of the molecule. The attraction is only between nearby amino acids, giving rise to the secondary structure. It can give rise to the beta pleated sheet and the alpha coil. This is the coil shown here. Here's the beta pleated sheet shown here. And then there's a third one, which I think the video never discussed. It can give rise to the random coil, where the coil can, randomly interact and respond to each other. The tertiary structure is the overall folding and the pattern that protein or polypeptide molecule makes. So you can call it the supercoil, how the protein winds around each other and forms the final, well, not necessarily the final, but the tertiary structure, okay? So the overall three-dimensional structure of the polypeptide chain is the tertiary structure. How you get this tertiary structure is from all of the interactions that you see in the secondary structure. And then one other, you can also have disulfide bonds, and I'll talk about that in a minute. But there is one difference, and that is all of these interactions on the secondary structure only happen between neighboring amino acid. In the tertiary structure, all of these interactions can happen all over the protein. For example, shown here, this amino acid near the end of the protein is hydrogen binding or being attracted to this amino acid near the start of the protein. So these are not neighboring amino acids. And then let's talk about the disulfide bridge. There is one attraction that happens in the tertiary structure that does not happen in the secondary structure, and that's the disulfide bridge. There we go, paint. Bring this out. So we have our protein, and it's just going to wrap around like this, go around something like that, come around like this, go around like that, and something else. We can have a cysteine group right here, which is the amino acid cysteine. and it has a sulfhydryl group on it. And then we can have the amino acid over here. Let me see if I can get another one. About right there. A little off, but that's close enough. Which has a sulfhydryl group over here. And I guess I should go HS first. Well, these hydrogens can come off. Let me see if I can remove that. Up. I can't remove it. So let me just erase that if I can. Up, wrong one. There we go. The hydrogens can come off, and then the sulfur can bond to each other. Let's just use the pencil. It's supposed to be a straight line. I'm not going to be able to draw that straight, but you get the idea. This link is a covalent chemical bond. It is very sturdy. How sturdy? When you get your hair permed, you put in sulfur disulfide bridges in your hair. And as you know, that can last in your hair for a month with showering. And if you ever were to just curl your hair using hydrogen bonds, which you put your hair in curlers and put bobby pins in and then put steam cap on and then put steam in your hair. My mother used to do this in the 1960s and 70s, and which I was very young in the 1960s, by the way. But he would make her hair curl, and then she would take her hair out of the curling iron and then take out the curls and the bobby pins, and then she'd have this nice curl. It's by hydrogen bonds. However, then she would go into the Oregon mist or the Oregon weather, and she lived in Portland or slightly outside of Portland, and the Oregon weather would have moisture in the air, and that would make her curls sag because each water in the air would bump into that hydrogen bond that she made in her curl, and it would break it. So the hydrogen bonds are very weak, and they can break just with the moisture in the Oregon and Western Washington air. Whereas the disulfide bonds make the curl in your hair, meaning the perm in your hair, which is very strong, and it lasts a month, even with showering. Any question about that? This link we can find in the tertiary structure of proteins. Like right there and right there. And it's actually shown right there and there too. Now, for any protein that's only made of one polypeptide, the highest level the protein can get is the tertiary structure. The quaternary structure is only seen in proteins which are made up of more than one polypeptide, such as hemoglobin and antibodies, which are made up of 4 polypeptides. Each of the polypeptides come together and then interact with other amino acids. So the amino acids on this polypeptide can interact with the amino acids on this polypeptide, and then form attractions and repulsions, making the final shape of the protein. And this we call the quaternary structure. And it's only seen in proteins that have more than one polypeptide making up the protein. And that happens in hemoglobin and antibodies. Any question about any of that? Let me check on when this class is supposed to end. I don't remember in the summer because it's a different schedule. Oh, we have until 6:50, so we have quite a ways to go. Any question about the levels of protein structure in a protein? With proteins, we also have globular proteins that take a roughly spherical shape. Hemoglobin takes this shape. And we have fibrous or thread-like proteins. And these are usually proteins in a structural molecule like collagen. We can have conjugated proteins, and these are combinations of amino acids with other organic or inorganic components. They are usually named by their non-protein component, like glycoproteins or a protein bound together with the carbohydrate molecule. Lipoproteins are a protein bound to a lipid molecule. Any question about that? Protein denaturation is an important topic. Proteins have an overall three-dimensional shape. And for the protein to function, it has to have its correct shape, its correct three-dimensional shape. If you change the shape of the protein, either by breaking hydrogen bonds or forming hydrogen bonds that shouldn't be there, you can get the protein to change its shape and undergo protein denaturation. When the protein is no longer functional, we say the protein is denatured. You can denature proteins by putting them in the incorrect temperature, the incorrect pH, or the incorrect salt concentration. A good example of a denatured protein is egg albumin. When you crack the egg and the egg white is liquid in the pan, it is a functional protein in the correct three-dimensional shape, assuming albumin is only a single protein. I don't know if it's has a quaternary shape or not, but the point is the protein is in its correct shape. It's acting as the protein should. You can denature it by heat. And when we cook our eggs and get the egg whites to be a solid egg white, that is denatured protein of the egg white. Any question about any of that? All right, let's move on to nucleic acids, the fourth of the biological molecule. There are two main nucleic acids, DNA, or deoxyribonucleic acid, and RNA, or ribonucleic acid. They consist of monomers called nucleotides. So all DNA and RNA is a bunch of monomers where the nucleotides are hooked one after the other. Nucleotides themselves consist of three parts. Let me blow this up. Get that out of the way. A nucleotide contains a 5-carbon sugar, shown here. And for DNA, it's deoxyribose is the sugar. For RNA, it is ribose is the sugar. And they're similar, it's just that there's an oxygen removed from, I think, deoxyribose. So that's the first part of the nucleotide. The second part is the phosphate group, and it's bound to the sugar. And then there's a nitrogen-containing base, and it too is bound to the sugar. There are in DNA 4 different bases which make 4 different nucleotides. We're looking at adenine here as one of the bases, and thymine here, that's another base. Any question about that? The bases are of two types. There are purines and pyrimidines. So these bases come as purines or pyrimidines, adenine or A, and guanine or G, and I don't care how you know it, either by A or adenine, are purines, and cytosine or C, and thymine or T are pyrimidines. Let me give you a trick for knowing your pyrimidines from your purines. For DNA, a pyrimidine has a Y in it, and cytosine and thymine, which are pyrimidines, all have a Y in it. So for DNA, you just look at the nucleotide, and if it has a Y in it, it is a pyrimidine. If it doesn't have a Y in it, it is a purine. Any question about that? Oops, I'm back up in the show. Now for RNA, we have three of the same bases, adenine, guanine, cytosine, but we do not have T, thymine. It is replaced by U, uracil. So if you remember that U replaces T, and T stands for thymine, which has a Y in it, then you'll know that uracil is a pyrimidine, because uracil replaces thymine, and thymine has a Y in it. Okay? And then the cytosine of RNA is also a pyrimidine, and the adenine and guanine are obviously purines. Any question about any of that? The nitrogen-containing bases pair with each other, meaning A will pair with T, or T will pair with A, and realize that if you are talking about RNA, then we replace T with U, and so A will pair with U. C pairs with G, and G pairs with C. That is how DNA is a double strand. The single strand of DNA is held together by covalent bonds, and it's held together by, let me see if I can blow this up. It's held together by the sugar here, held to the phosphate, held to the sugar. So it's the sugar phosphate backbone, which holds a single strand of DNA together. The double strand of DNA is held together only by the hydrogen bonds between the complementary bases. And the bases do not hold the single strand of DNA together. They're sort of the steps on the spiral staircase. And G pairs with C. It forms three hydrogen bonds, G and C. A and T form two hydrogen bonds together. Any question about any of that? And yes, DNA wraps around itself like a spiral staircase. The hydrogen bonds are very weak, and in our body temperature, this hydrogen bond here breaks and reforms all the time. But DNA is held together because there's lots of hydrogen bonds holding the DNA together. And even though this one breaks, it's held together by this one and these, and then this one will break and that one will reform, holding the DNA together. Any question about any of that? Okay, so DNA is a double-stranded molecule. The single strands are held together by covalent chemical bonds. The double strand is only held together by hydrogen bonds. The sugar is deoxyribose. All right, any question about any of that? RNA, the sugar is ribose. RNA is not a double-stranded molecule, at least in our cells. RNA is a single-stranded molecule. It too is held together by the sugar phosphate backbone. The bases are, of course, A, G, C, and then uracil U replaces T. There are three major kinds of RNA: transfer RNA called tRNA, messenger RNA or mRNA, ribosomal RNA or rRNA. So when a gene is being transcribed, how does the polymerase know to make tRNA, messenger RNA, or rRNA? How does the polymerase know which RNA to make? Anyone? You guys don't like guessing. The polymerase doesn't have to decide. The polymerase is transcribing the gene, and the gene will either code for messenger RNA, tRNA, or rRNA. If the gene is an rRNA gene, then it'll be transcribed as rRNA. If the gene is a tRNA gene, then it'll be transcribed as tRNA. If the gene codes for a protein, it'll be transcribed as messenger RNA. The only difference between tRNA, messenger RNA, and rRNA is the sequence of the nucleotides. So they'll have a different sequence. Any question about any of that? All right. All of these different types of RNA are important in protein translation. ATP is also considered a nucleic acid. ATP is adenosine triphosphate. It is a molecule shown here having 3 phosphate groups attached to it. It does have ribose as the sugar, and then adenosine as the base. Let me see if I can blow that up. It does have 2 phosphate groups which are bound together by a high energy bond. So wherever there's a squiggly line shown right here, that's a high energy bond, and that one is too, a high energy bond. If the cell needs energy, most of the time it'll break this high energy bond, and then that will release a lot of energy, and the cell will use that energy for what the cell needs energy for the work that it's doing. If the cell needs additional energy, it can then break off this phosphate group, and that too will deliver a bunch of energy. So normally when the cell needs energy, it gets it from ATP. ATP is the primary energy cell source for our cells. We call it the energy currency because currency is what we use to buy things in life, and ATP is what cells use when they need energy. They'll break ATP to get the energy they need for whatever they're doing that needs energy. Any questions about ATP? All right. ATP is made by dehydration synthesis. You simply get ADP, adenosine with 2 phosphate groups, combine a 3rd phosphate group, and that's an inorganic phosphate group, meaning it's just phosphate, which is inorganic. And then you add energy, and then it will form water and ATP, adenosine triphosphate. So to make ATP, it takes energy, and then if the cell needs energy, it breaks that phosphate off from ATP by condensation reaction. Is that right? No, by hydrolysis, sorry. By hydrolysis, it adds water, breaks off that phosphate. The water splits, part of it will go to the phosphate here and the other part to the phosphate here. And that will release energy, and the cell will use this energy for its energy needs. Any question about any of that? Okay, what molecules that we discussed today have hydrogen bonds? And the hydrogen bonds give the molecule important properties. That's a question. I want you guys to answer it.
Well, the
Sure.
nucleic acids, DNA and RNA.
Yeah, they have hydrogen bonds. How about proteins? Do they have hydrogen bonds? Anyone? Okay, you guys need to think about this. And then you can think about what would happen to these molecules if the hydrogen bonds were all to disappear. Another molecule that we talked about that has hydrogen bonds is water. And as you know, if in liquid water, all of the hydrogen bonds were to disappear, we no longer have hydrogen. I mean, we never longer have liquid water. The water would be gaseous if all of the hydrogen bonds were to disappear. All right, any questions for chapter 2? If not, I'm going to stop chapter 2 here and begin chapter 3. We'll just get started with chapter 3. So this is a slide showing you the, oh fudge, forgot about that. An upgrade to Windows on this computer, and now that's always going to show up. Chapter 3, observing microorganisms. I'm showing you the major goals and a rough outline. As always, know the terms. To understand the size of organisms and the properties of microscopes. Be able to understand the major differences between a light microscope and the electron microscope. 3, understand why staining improves resolution. 3A, know the 3 types of stains. There are simple stains, differential stains, and special stains. And then we're going to talk about the gram stain. Understand the gram stain, its procedure, and the basis of its differential staining. Any questions? All right. In this lesson, you need to know the metric system. And when you're working with the metric system, let me see if I can get that going. Come on. You need to convert between one metric unit and another metric unit. You should know a kilometer, be able to convert that to a meter. You don't need to know a decimeter, but you should be able to convert a meter to a centimeter, and a meter to a millimeter, and a meter to a micrometer. And the micrometer is the size that a light microscope sees. And then a nanometer is the size that an electron microscope sees. So you should know a nanometer. You do not need to know a picometer. The meaning of the prefix is a kilo means 1,000. So there are 1,000 meters in a kilometer, and that's shown here. The meter is the basic standard of unit in metric for the length of something. The centimeter, we have 100 centimeters equal 1 meter, or 1/100th of a meter equals 1 centimeter. A millimeter is we have 1,000 millimeters in 1 meter. A micrometer means we have 1/1,000,000th or a million micrometers in a meter. Okay, any questions about that? So you need to know the meaning of the prefix. You should know the meter, and then you can change what you're measuring, like for weight or mass, we use gram. And you use the same prefixes, like there's a kilogram and there's a centigram. And then what's the other one? Liter, for measuring liquid volumes. We have a liter, so that's the standard. And you use the same prefixes there. What's nice about metric is everything's on a base of 10. You don't have 12 inches in a foot, for example. Everything is 10, makes up the next one. You do not need to convert between metric and English and metric. I'm not going to ask you to know that, but you should know metric. Any question about that? You are expected to know metric for quizzes and exams. I think the first quiz on chapter 3 will give you one question where you need to convert between one metric unit to another. The next slide is showing you one of the reasons why you should know the metric system. This map is showing you all of the countries in the world that do not use the metric system. And if you notice, it's only 3 countries in the world. Any question? And the United States is the only country that is a big country that does not use metric system. If you don't know, the United States is slowly moving over to using metric. And that's why on our rulers, you can find metric in English on most rulers. And on the cars, you can find metric in English on the, what do they call it, the speedometer. And then on many signs, not in all states, but in many states, you can find both metric and English speed limits given. And that's a good thing because I remember my brother was first traveling in Canada, and it told him to drive so many kilometers per mile. And he said, "What speed is that?" Unfortunately, I was pretty good in math and just had learned it in a year or two earlier. And so I could remember that let me see if I get this right. 1 kilometer is 0.6 miles. And so I could give them the approximate speed limit. The other reason why you should know metric is it is the units that are used in science and medicine. So you should know metric for these 2 reasons. Mainly because everybody in the world, except for the United States, and I think that's Liberia, and I don't even know that country, maybe Burma, are the only countries in the world that do not use the metric system. The United States is slowly transitioning over to use the metric system, and it's been a very slow process. We began the process when Thomas Jefferson was president, and as you can tell, we haven't gone very far to using metric. And then science and medicine use metric system. Oh, for your practice on using the metric system, I do have some quizzes which you can use to practice on using the metric system. I think it's under lab, no, it's under lecture week 3, metric quiz 1. You need to open this. And it won't work here because it will not work inside a Canvas. But if you click here again, it'll then open it on another page and it'll work. In this case, I think we hit start to get the quiz to work. So select the base unit of the metric system for length. It is meter. Correct. Shows you the answer nice in green. If you take another question and it's wrong, for volume, and I go meter again, it'll say incorrect, try, good try. The correct one is actually liter. Oh, it shows you the correct answer too. in green. All right, any questions about any of this? If not, I think I'm out of time. So I'll see you at 7 o'clock for the lab. Any questions? All right, bye.